the human embryo begins its story at the formation of the zygote at fertilization. This is a single cell structure that grows and grows and grows until it becomes a whole organism. This occurs at birth. The human embryo is peculiar because there are debates as to what its nature is. Is the human embryo human, or is it subhuman? If it is human, the killing of a human embryo then becomes murder. If it is not human, then what is it? At what point does it become human? There are several theories, and the theories are as follows. We have the conception theory, which is the absolutist theory, and this is the most widely accepted. It is strange, though, that it is the latest model of understanding of the human embryo, having only been used in the 19th century because of the discovery of the human egg by Bonvea. Before this, several models were followed, including the ones used by the Jews and the Roman Catholic Church, which says the embryo becomes human only when it reaches the fetal stage, at which time it will have acquired its appendages so that it's recognizable as human. That is, it will have a head, it will have limbs. But before then, it was not considered human. This was because the human egg was not recognized as an entity involved in fertilization until the discovery by von Baer in the 19th century. The advantage of a theory of conception is that it has science behind it. Since the very first process of life begins at fertilization, which is the same point of conception, and there is no other process which separates new life from maternal life except this one. It is therefore difficult to expect scientists to believe that life begins before this. Strangely, though, it is even the scientists themselves, by that I mean investigative scientists, who refuse to accept the theory of conception. Most other people who may be shark scientists or who may have little knowledge of biology support this theory enthusiastically. The next theory is the morular theory. This theory has the least of support. It is found mostly amongst modern reproductive technologists who have discovered that maternal transcription of DNA, which controls the early embryo behavior, continues until sometimes 4-cell or even morular stage of the embryo, when the actual zygotic or embryo transcription of the embryo genome begins. When the later begins, the proteins produced for the physiology of the developing embryo are strictly derived from the transcriptional processes of the nucleic acids of the embryo itself. In that case, the embryo now becomes independent and fully human. The brainwave theory suggests that it is only when the brainwave appears, at which point it is assumed that the embryo has now developed a functional brain. That is the time the embryo becomes human. The soul theory says it is only when the embryo acquires a soul that it becomes human. Most Islamic theologians would suggest that this date will be 40 days after fertilization. Primitive streak theory. This theory suggests that the organ primitive streak appears at about the 14th day after fertilization. The primitive streak is unique because it brings organization to previously unorganized embryo. Before the formation of the primitive streak, the embryo does not have head, tail, or limbs, but just a ball of cells. But after the appearance of the primitive streak, it now has presumptive regions. The nervous system theory, which is the next theory, suggests that an embryo cannot be said to be fully human until it develops a nervous system. This theory relies heavily on the principle that without a nervous system, the organism cannot feel pain. And if there is no pain, then there can be no actual life. In that case, the embryo is considered to be a plant. It has the status of a plant because plants don't have pain.
Hold your leg and push. You are trying, but push harder. Harder. Harder now. What are you shouting for? Oh yeah. Yeah. You are trying. Put your buttocks down and then push. Oh yeah now. Are you having pain now? Yes. Go. You know, she just do slow and stop. Are you having pain now? Or you are doing one hard one, like hard. Don't stop. Push. This one. Oh yeah now. This episode told me be like that. Be like that too. What are you shouting for?
Don't waste that time.
Don't waste the pen shouting. Push. Push. Yeah. Do it. Oh yeah. Push now. Oh yeah. Push. Push. This one. Good. Push now. Push. Oh yeah, now push. Madam, now push. Well done, eh? Oh yeah. Hold on.
You can't go down.
Oh yeah, continue. Oh yeah.
Well done.
Try. Give us the name of the give it to hold it now. Oh, sorry. That's what I wanted to do. I forgot. Madam,
The secondary oocyte with a single polar body is released from a mature Graafian follicle. It is taken up by the fimbriae of a particular fallopian tube and migrates into the ampulla of the tube. There it meets numerous sperm cells already released for fertilization, or it may wait for not more than 3 days for the sperm to arrive. The sperm cells attempt to penetrate the egg, but only one succeeds. Male and female pronuclei then fuse to cause fertilization. Cleavage begins with the zygote forming the first cleavage division, leading to 2 daughter cells. From 2 cells it goes to 4, and then 8, and then 16, at which point it is called morula. It proceeds to 32 and then 64, after which cavitation occurs and the embryo becomes a blastocyst with two populations of cells, the inner cell mass and the outer cell mass. Further development leads to the inner cell mass, which develops into the actual embryo to become the germ disc, initially with only one germ layer called the ectoderm, but later joined by the endoderm derived from the same ectoderm. Growth of the embryo is quite rapid. Between the 4th and the 8th week of gestation, there is massive proliferation and differentiation of cells to form various organ systems. At the end of the 8th week, almost all the primordia of various organs and systems of the body are already formed. Process for formation of these organ systems is called organogenesis. In the early fetal period, there is rapid growth of the fetus until the late fetal period, which is characterized by increase in fetal weight. From 26 weeks of fertilization to 1 month after birth, it is called the perinatal period. Perinatology is the branch of medicine that deals with the study of fetus in the perinatal period. Perinatal medicine therefore combines some bits of obstetrics and pediatrics. During the perinatal period, amniotic fluid can be obtained from the amniotic cavity by a process called amniocentesis, and the fluid is subjected to various analyses for the diagnosis of various disorders. For example, amniotic cells can be cultured and chromosomal stages can be done to detect disorders like Down syndrome, Turner syndrome, hemophilia. Alpha-fetoproteins can be detected in the amniotic fluid, and therefore it is known to increase in a condition like spina bifida cystica. Intrauterine blood transfusion can be performed during perinatal period, and ultrasound or ultrasonography can be employed to detect conditions like anencephaly, fetal ascites, spina bifida, intrauterine growth retardation, etc. Estimation of gestational age or fetal age is done by the following: information on last menstrual period, LMP, fertilization age, measurement of height such as crown rump length, measurement of biparietal diameter, somite count, etc. shortly after the formation of the intraembryonic mesoderm, the embryo folds laterally and anteroposteriorly in order to reduce the size of the gut formed from the yolk sac, and also to place the intraembryonic xylem ventrally. Embryo folding. The flat trilaminar embryonic disc progressively increases in size and undergoes folding to be transformed into a cylindrical structure. Embryo folding takes place in two planes: longitudinal folding and transverse folding. These foldings take place in a vertical direction. During longitudinal folding, a head fold and a tail fold are formed. As the head fold forms, part of the yolk sac is incorporated into the embryo from the primitive foregut. This primitive foregut is separated from the stomatodium by the oropharyngeal membrane, the cardiogenic area, together with the primitive heart tubes and the septum transversum. They come to lie ventral to the primitive foregut, with the septum transversum lying caudal to the primitive heart. The septum transversum takes part in the formation of a diaphragm. As the tail fold forms, part of the secondary yolk sac is incorporated into the embryo as the primitive hindgut. This primitive hindgut is separated from the cloaca by the cloaca membrane. During transverse folding, part of the secondary yolk sac is also incorporated into the embryo as the primitive hindgut. This primitive hindgut is separated from the cloaca by the cloaca membrane during transverse folding. Part of the secondary yolk sac is also incorporated into the embryo as the primitive midgut, and as this primitive midgut forms, it becomes attached to the dorsal aspect of embryo by the dorsal mesentery. As a result of a transverse and longitudinal folding, the embryo becomes attached ventrally with the remains of the secondary yolk sac. The yolk sac and the connecting stalk all are formed, and they lie close and ventral. Together they take part in the formation of the umbilical cord. The embryo initially forms two germ layers, the ectoderm and the endoderm. The embryo is therefore called the bilaminar embryo. Later, a third germ layer is added. The process for the adding of the third germ layer is gastrulation. This process leads to the organization of the embryo so that the embryo now has pores. It is organizable to head, tail, or limb boards. The primitive streak helps in the formation of this third germ layer. Cells that are coming from the ectoderm insinuate themselves between the ectoderm and the endoderm around the primitive streak. They migrate rapidly around the whole of the embryo and assist the process of organization. Somites reach up to 42 to 44 in number when they are fully developed. They are formed from paraxial mesoderm and are divided into dermatome, sclerotome, and myotome. Dermatome migrate into the skin to form the mesoderm of the skin, mainly the dermis. The myotome forms muscles of the trunk region. The skeleton forms the cartilage that forms the bone of the vertebrae. The lateral plate mesoderm begins to develop spaces within it, and these spaces coalesce to form the intraembryonic xylem. The intraembryonic xylem surrounds the cardiogenic area, which forms the pericardium, and other coelomic cavities within the body wall. Intraembryonic xylem divides the lateral mesoderm into two parts. The one that continues with the extraembryonic mesoderm surrounding the amnion is called the somatic intraembryonic mesoderm, and the one that continues with the extraembryonic mesoderm surrounding the primary yolk sac is called the splanchnic extraembryonic mesoderm. The somatic intraembryonic mesoderm together with the overlying ectoderm is called the somatopluric mesoderm, and this forms the body wall. The splanchnic intraembryonic mesoderm and the underlying endoderm form the splanchnopluric mesoderm, and they are responsible for covering the wall of the gastrointestinal tract. The intraembryonic xylem forms three main body cavities. Number 1, the pleural cavity. Number 2, peritoneal cavity. Number 3, pericardial cavity. There are three processes involved in the development of the face, and they are the frontonasal process, the maxillary process, and the mandibular process. The frontonasal process becomes the forehead and the nose. The maxillary process forms the face below the eyes. The mandibular process forms the face below the mouth. As the development of the face continues, the nasal cavity is being formed. The brain also expands. At the pre-somite stage, the face consists of only a bulging forehead superiorly and a developing pericardium inferiorly. Laterally, we can see the mandibular process. The stomoidal membrane covers the opening into the mouth. Later, the membrane breaks down so that there is a communication between the mouth and the developing pharynx. As development continues, the mandibular processes on the right and the left form the maxillary process. As the mandibular processes continue their development, they will now fuse at the midline, forming the lower jaw together with the lower lip. Superior to the mandibular process and the stomatodium, but medial to the maxillary process, is the olfactory placode. This placode will develop into a frontonasal process. The process consists of two main structures, medial and lateral nasal folds. The area between the medial and lateral folds is therefore developing into the olfactory system. As development continues, the lateral nasal fold fuses with the developing maxilla, forming the naso-optic furrow, which enters into the developing eye, placed above the frontal process, that is, the frontonasal process. The medial and the lateral nasal folds will meet and fuse, forming a space between them, which becomes the anterior nares. This will be the opening into the nose. The right and left medial folds then come together, reducing the space between them. This space is going to become the nasal septum internally. The maxilla also fuses with the medial nasal fold, and the right and the left maxillary processes will meet the frontonasal process at the midline, so that the two will form the upper lip, while the maxilla alone will form the upper jaw. The maxillary and mandibular processes now contribute to the formation of the cheeks. A transverse groove separates the bridge of the nose from the developing forebrain, which then becomes the forehead. The mesenchyme of the nose forms ossification centers for the ossification of the two nasal bones. Finally, the muscles develop from the circumpharyngeal arch will invade the face region and the auricle to form the muscles of facial expression, which maintain their nerve supply from the facial nerve. The key to the understanding of the anatomy of the head and neck region lies in the understanding of the development that takes place in the pharynx. And this includes the development of the arches, the pouches, and the clefts. Almost all the features in the head and neck region develops from the pharynx in a model similar to the gill clefts of fishes. Even the nerve supply to these primordial structures are derived from head region, even if they later migrate to neck or other regions. Most of the major organs are preformed in pouches, pharyngeal pouches. The original head does not contain these structures. It has only the brain and a developing eye. Pharyngeal arches are responsible for the formation of the structures derived from mesoderm of the head and neck region. Their development leads to increased separation of the space between the stomatodium, which is the primitive mouth, and the pericardium containing developing heart. Also, they are placed lateral to the developing brain. The lateral plate mesoderm is responsible for the formation of a pharyngeal mesoderm, and this mesoderm will differentiate in the following: the cartilage and muscle. The cartilaginous elements later ossify to become bones, while the muscles are derived from inside to differentiation of arch, mesoderm or mesenchyme. Other structures which are associated with the arches invade the developing mesoderm of the arches. The structures which supply the premandibular mesenchyme leads to the formation of extraocular muscles, and they include the superior, medial and inferior recti together with the inferior oblique and the levator papillae superioris. The maxillomandibular mesenchyme leads to the two muscles, the lateral rectus and superior oblique. The nerve to the maxillomandibular mesenchyme includes the trochlear nerve and the abducens. The mandibular arch gives rise to muscles of mastication, the temporalis, the masseter, the medial and the lateral pterygoid. The mandibular cartilage forms the Meager's cartilage, the quadrate cartilage, the mental ossicles, anterior ligament of the malleus, and the sphenomandibular ligament. The nerve to the mandibular arch is the mandibular nerve, which is a branch of the trigeminal nerve. The hyoid arch is the second arch. It gives rise to muscles of facial expression. The nerve supply to the hyoid arch is the facial nerve. The third arch gives rise to the stylopharyngeus. The nerve to the stylopharyngeus is the glossopharyngeal nerve. The fourth pharyngeal arch gives rise to the cricothyroid muscle. The nerve supply to the cricothyroid is the superior laryngeal branch of vagus nerve. The sixth arch gives rise to intrinsic muscles of the larynx. The sixth arch nerve is the recurrent laryngeal branch of the vagus nerve. The caudal arches give rise to the muscles of the pharynx, such as the superior constrictor, inferior constrictor, and the middle constrictor. Nerve supply to the caudal arches are from the cranial accessory through the vagus nerve. The development of the tongue is close to the development of the arches. The pouches, pharyngeal pouches, are endodermal in origin. They initially communicate with the pharynx, but later this communication narrows, and then they form pouches, which have ventral and dorsal recesses. The tongue does not allow the formation of the ventral recess in the first pouch. On each side of the dorsal recesses of the first and second pouches, there is an expansion of the pouches to form the tubotympanic recess with a single narrow pharyngeal opening. The ventral recess of the second pouch between the soft palate and the tongue forms the palatine tonsil. The right and the left ventral recesses of the third pouch form the thymus gland. The dorsal recess of the third pouch forms the parathyroid gland, or parathyroid 3 or inferior parathyroid. The dorsal recess of the fourth pouch forms the superior parathyroid gland, or parathyroid 4 or superior parathyroid gland. The last pouch, which is called the ultimobranchial body, forms the pouch and releases cells which are called C cells. These cells invade the thyroid gland and produce calcitonin in the adult. At the third week of gestation, the heart develops from fused endothelial tubes, forming the bulbus, the ventricle, and the atria. The atria begin to rise above the ventricles. The truncus arteriosus is formed, leading to the formation of the aortic sac above the bulbus cordis. The heart was originally a single chamber, but as development continues, it becomes partitioned into 4 chambers, 2 atria and 2 ventricles. The ostium primum is the opening between the left and the right atria. Later, a septum secundum is formed so that the septum primum joins the endocardiac cushion, leading to the formation of a septum secundum, which later becomes converted into foramen ovale. The foramen ovale closes up at birth so that blood now runs from the right atrium to the right ventricle. The interior of the heart. The single-chambered heart soon gives rise to the formation of the endocardiac cushion in the middle, with a still wide communication between the atria and the ventricles. Septum primum is formed at the roof of the right atrium, which approaches the cushion and forms the ostium primum between it and the cushion. It then becomes detached on the top, with the formation of the ostium secundum behind it, and the obliteration of the ostium primum as it joins the endocardiac cushion. The ostium secundum then becomes the foramen ovale at the mature 6 weeks heart. With the formation of the atrioventricular valves between the atria and the ventricles, the heart becomes 4-chambered, with only the foramen ovale as the opening between the atria. There are 2 connections from the right side of the heart to the left in the fetus. One is via the foramen ovale, and the other one is via the patent ductus arteriosus. At birth, these two foramina close off. Fetal circulation. Blood in the fetus flows from the posthepatic inferior vena cava to enter the right atrium. It is separated from the blood coming from the head and neck region, which pass straight into the right atrium via the intravenous tubercle. So that blood from the SVC goes straight into the right ventricle, and the other goes through the foramen ovale to the left atrium. The right ventricular blood then enters the pulmonary trunk to be shunted via the patent ductus arteriosus to the aorta, from where it is passed to the rest of the body and to the placenta. Blood which enters the left atrium is immediately passed into the left ventricle, and from there to the ascending aorta and then systemic circulation. This fetal circulation is designed to shunt blood from the right to the left, so that blood is diverted from the immature lungs to the placenta through systemic circulation, where it is oxygenated via the umbilical arteries and then veins. In the outward circulation, the right to left shunt is corrected. This is done immediately after the birth of the child. The ductus arteriosus and foramen ovale close. So also is the umbilical circulation. Blood now goes into the lungs and comes back via the IVC and the SVC. There are 6 aortic arches, but the 5th is transient. The aortic sac develops from the truncus arteriosus and is connected to all the arches. Then the development shapens the arches and they form the aortic sac superiorly. The right dorsal aorta of the 4th arch moves upwards, and the left dorsal aorta also moves downwards and forms the final arteries in the embryo. Take origin from the aortic sac, which originally was connected to a paired dorsal aorta. They arise segmentally to supply the pharyngeal arches as 1st, 2nd, 3rd, 4th, 5th and 6th aortic arches. The 5th aortic arch disappears with the 5th pharyngeal arch. Again, the 1st and the 2nd arches are obliterated at their ventral aspects. The 3rd arch is incorporated in the formation of the common carotid arteries. The 4th aortic arch, together with The cardinal, vitelline and hepatocardiac veins develop below the heart. They form series of longitudinal anastomoses, which later come together to form the definitive inferior vena cava. The ductus venosus is also formed above the liver. It passes through the liver and is connected to the subcardinal anastomosis initially. The inferior vena cava also becomes formed and is connected to the ductus venosus.
Veins are not segmental the way arteries are in the embryo. The original veins are the paired anterior and posterior cardinal veins, which form the common cardinal veins on both sides. They enter the sinus venosus. The right anterior cardinal vein forms the right internal jugular vein and the brachiocephalic veins, and also part of the superior vena cava above the azygos vein. Across anastomosis between the anterior cardinal veins form part of the brachiocephalic vein of the left. Posteriorly, the subcardinal veins appear and they drain the mesonephric ridges. The body wall is drained by the supracardinal veins that appear later. The inferior vena cava is formed from many veins. The initial ductus venosus is formed within the liver before the inferior vena cava. The lungs develop from a single diverticulum forming the laryngotracheal tube, which divides and subdivides until it becomes the actual lung.
Midgut herniation begins about the 4th week. It then returns after rotating with the duodenum passing above and forming the C curve. By the time it returns, the duodenum is very well formed. The whole of the gastrointestinal tract forms from the yolk sac as a result of the carving in and the folding of the embryo. The stomach tube is formed and it enlarges. The pancreas is also formed as diverticulum, 2 diverticula from the gut tube, 1 dorsal and 1 ventral. With rotation, the gut rotates, the stomach rotates, and the 2 ventral and dorsal pancreas will fuse. At the same time, the gallbladder is being formed. The liver is also being formed within the septum transversum. The stomach rotation leads to the formation of the augmented bursa behind the stomach. As the liver is being formed, the visceral veins that pass through it are also formed. They are 3: umbilical, vitelline, and hepatocardiac. They change the anastomosis, forming the subdiaphragmatic anastomosis, duodenal anastomosis, which surrounds the duodenum, and with further recanalization and the liver expanding, these vessels obliterate so that the umbilical vessels become completely obliterated. The ductus venosus is now formed. And as the development continues, the ductus venosus increases in size, the porta vessels develops, and the duodenum develops also with the degeneration of vitelline vessels. With further development of the ductus venosus, the inferior vena cava is formed. The superior vena cava also forms superior.
The kidneys are formed from three important preceding structures. The first is the pronephros. It is not very clear whether this exists in the human embryo, as it does clearly in those lower animals. But if it does, it stays for a short while and may occur in the intermediate mesoderm, which is opposite the cranial somites. It gives way to the mesonephros, which develops tubules and extends from the septum transversum to the third lumbar vertebrae. Its long duct is then called the mesonephric duct or Wolffian duct. The next renal system is the metanephros, which forms below the mesonephros at the sacral region. A ureteric board then grows out of the caudal part of the mesonephric duct and grows into the metanephros. It consolidates itself and becomes the definitive kidney, complete with circulatory and tubular parts.
The kidney then rises from the pelvis and moves to the abdominal cavity. With it, also the ureters. The urinary bladder is also formed from the urogenital sinus. In the female, the ovary develops by the invasion of the germinal epithelium by primordial sex cells. The sex cords are then formed. The mesonephric duct degenerates, but the Mullerian duct persists, and the Mullerian duct forms most of the ductal systems of the female reproductive system. In the male, at 6 weeks, the sex cells, the primordial sex cells, begin to invade the germinal epithelium and also form sex cords. There is now a connection from the mesonephric duct to the tubules. These tubules form the seminiferous tubules, while the Mullerian duct degenerates. Primitive duct system are laid down at 4 weeks of gestation, with the formation of the primitive bladder and the urogenital sinus. Later, the mesonephric and the paramesonephric ducts are formed together with the Mullerian duct. As penis is formed, the urogenital sinus begin to differentiate and form a definitive bladder together with the urethra. Later, the duct system begins to form the barnaculum, forms of the mesoderm, and leads to the anchor of the testis. The center of the testis then follows. It goes gradually down to the scrotal sac. The descent of the pelvic viscera now follows.
Primordial sex cells are on the yolk sac in the rhizomite embryo. They migrate from this position to the medial aspect of the mesonephric ridge. The epithelium of the mesonephric ridge then proliferates and forms its own ridge called the genital ridge. Two ducts are now recognized as the Wolffian or mesonephric duct and the paramesenteric or Mullerian duct bilaterally. The mesenteric duct is associated with the mesenephros developing kidney system. In the male, as soon as the sex cells arrive, the epithelium of the genital reform sex cords, which becomes cut off from the surface epithelium and are linked to the mesenephric tubule system from the primitive seminiferous tubules. Swelling and genital folds. The genital tubercle becomes the phallus. In the male, it continues its development on it until it is fully grown and forms the penis, with the penile urethra open at its tip, that is the tip of the penis. In the female, it goes for a little while and stops for the growth to allow the urethra to develop outside walls in the anterior wall of the vagina. In both male and female, it leads to the formation of similar phallic structures such as the corpora cavernosa and the corpus luteosum. In the female, the Mullerian tubercle becomes the hymen and moves to the surface. The genital fold then becomes the labia minora, while the genital swellings become the labia majora. In the male, the genital tubercle becomes the penis, genital swelling scrotum, and genital fold the penile urethra. The development of the limb begins from the limb board. After organizational influence of the primitive streak, the structures in the limb differentiate in situ, most especially the mesenchyme, which may be derived from mesenchyme of the body wall, which is the somatoplastic mesenchyme. The muscles are not formed pyotomes, but differentiated within the limbs and are penetrated by the nerve and blood supply. After then, anywhere they migrate into, they carry along the vessels and nerves, and their migratory movement leads to fusion, extension, and recombination of the nerve fibers, hence the formation of plexus in the limbs. Bones ossify in three ways. They may ossify in membrane, and then they will be called membrane bones, or they may ossify in cartilage, then they will be called cartilaginous bones. A third category stands alone and affects the clavicle, in which ossification is taking place in the pre-cartilage. Usually, you have a primary ossification center for every bone, with many secondary ossification centers, which then spread the wave of ossification around the entire bone until it is completed. The central nervous system begins as a single tube with wide openings at the ends called anterior and posterior neuropores. But as development continues, they form what are called flexures. Two are ventral, whilst one is dorsal. The ventral ones are cervical flexures between the spinal cord and the medulla, and the mesencephalic flexure placed in the midbrain. The dorsal flexure is the pointing flexure. Three primary brain vesicles are now recognizable. They are forebrain, midbrain, and hindbrain. They undergo considerable distortion and flexions until they reach their level of maturity.
The nervous system is concerned with impulse generation and transfer. It obtains impulses from the environment using sensory apparatus known as receptors. The receptors acquire the impulse and pass them to the central nervous system. We have two morphological divisions of the nervous system as follows: the central nervous system and a peripheral nervous system. The central nervous system has two main organs, brain and spinal cord. The peripheral nervous system consists of all peripheral nerves, which may be either motor, sensory, or mixed. The nerves associated with the peripheral nervous system can be divided into two types: spinal nerves and cranial nerves. If the nerve is a spinal nerve, it goes to the spinal cord. If the nerve is a cranial nerve, the central process goes to the brain. A ganglion is a collection of cell bodies outside the central nervous system. A nucleus is a collection of cell bodies within the central nervous system. A nucleus is a collection of cell bodies within the nervous system, but a nerve is a collection of nerve fibers outside the central nervous system. A nerve fiber is an axon. A tract or a fasciculus is a collection of nerve fibers in the central nervous system. A funiculus or column is a collection of fasciculus. A funiculus or column is a collection of fasciculi or tracts. There is also a functional division of a nervous system. This is divided also into two types: the somatic nervous system and autonomic nervous system. Somatic nervous system takes care of the voluntary system of the body, while the autonomic nervous system takes care of the involuntary system of the body. autonomic nervous system is further divided into two types: a parasympathetic system and a sympathetic system. The spinal cord has 31 nerves. They are all called spinal nerves. Motor nerves are provided with multipolar neurons as opposed to the pseudo-unipolar neurons of the sensory nerves. Multipolar neurons have only one axon and several dendritic processes. Their cell bodies lie in the central nervous system. Cervical nerves have 8 nerves from C1 to C8. Thoracic has 12 from T1 to T12. Lumbar has 5 from L1 to L5. Sacral has 5 from S1 to S5. The coccygeal has only 1 nerve. Nerves associated with the brain are called cranial nerves. There are 12 pairs. You have the first nerve, which is the olfactory nerve. The second is the optic nerve. The third is the oculomotor nerve. The fourth is the trochlear nerve. The fifth is the trigeminal nerve. The sixth is the abducens nerve. The seventh is the facial nerve. The eighth is the vestibulocochlear nerve. The ninth is the glossopharyngeal nerve. The 10th is the vagus nerve. The 11th is the accessory nerve. The 12th is the hypoglossal nerve. Cranial nerves are motor, sensory, or mixed. They also have autonomic nerves, which are usually parasympathetic. There are 4 parasympathetic ganglia in the head and the neck region. They are as follows: ciliary ganglion, pterygopalatine ganglion, otic ganglion, submandibular ganglion. The brain itself is divided into 2 main parts. There is the brainstem and cerebral together with the cerebellar hemisphere. The brainstem consists of medulla, pons, midbrain, and diencephalon, which is usually referred to as the higher brainstem.